There's the idea of that perhaps the amount of ketones concentration are a little more tied to your brain to body weight ratio. So smaller people, the brain size is pretty constant in a lot of cases. So, smaller women may have higher concentrations than bigger men because the amount of water that those ketone bodies have to be dumped into is bigger for a man, so it's a lower concentration, versus a woman, it's smaller volume of water, so it's a higher concentration, even though they have the same brain size.
Interesting. I've never heard that before.
Yeah,
that makes so much sense, though.
Yeah, it's kind of if it's being used for fuel for the brain, then that's kind of where that concentration. But that's a longer term. If you were to do a 30-day fast, you may start to see some differential there. And there's a gentleman who's passed away, but he did a lot of this fundamental research.
Cahill,
yeah. And he had a paper where he kind of showed that children typically would have higher concentrations than women than men, and you'd have that kind of order, and that makes sense for children, right? Because they have the brain to body ratio is smaller, or is higher than you would for a man.
Hey friend, welcome back to episode number 245. So if you've ever wondered what it means to be fat adapted, what it means to be in nutritional ketosis, and how to assess if your body is fat adapted or keto adapted, this is a show for you. Joe Anderson, literally, he breaks down every metabolite, how we test this, how we assess this, and ways to get into ketosis and become more fat adapted. It's an awesome show. He's a chief science officer over at Levl. It's a great device that has an app associated with it. I've been experimenting with this since early April, late April. It's a great tool. I recommend it. Links are below this if you want to learn more about it. And this show is brought to you by our friends over at perfectketo.com, makers of high quality exogenous ketones and MCT oil formulations, and they have a new great product. It's actually a low carb, high fat bar that you can take on the road, like when you travel, when you're at meetings and you can't really go get the meals that you would like, or you're very busy with life. This is a low carb, high fat bar made from real whole food. The links are below this video, and as a loyal high intensity health listener and subscriber of our channel, you can save over 20% off on your first order. Just go to perfectketo.com/HIH. Again, that URL is perfectketo.com/HIH, and at checkout, be sure to enter in the promo code HIH to save your 20% off. Let's dive back into it with Joe, and I sincerely hope you enjoy the show.
Rewind the clock a little bit. So you're working with Levl now, helping them out with this ketone meter that looks at breath acetone, but you had a research interest in breath acetone for other applications, and so you've been interested in studying like volatile acids and stuff like that through breath for a while, huh?
That's correct. Yeah. So I got a PhD, and the topic was how do chemicals come out in breath? And so the chemicals we were interested in, one of them was kind of the class of chemicals that acetone would fall into. And the whole focus there was really to understand lung health. So it wasn't so much to understand metabolism or what's going on in the body. It was more of if the chemical comes out in a certain way, we can tell if you have maybe diseased lungs. That was one of the hopes. And also just understand basic biology better.
Yeah.
Yep.
And how was acetone the first metabolite you guys were looking at, like back in the day? Or?
That was the first one we looked at because what my goal is to use something that's naturally produced that then we could potentially take into a clinical setting to understand lung health. We had put chemicals into the body and watched how they come out to understand lung health, but obviously that takes another step. You're infusing something into the blood, watching it come out. That's fairly invasive. Yep.
So when acetone is formed, is it mainly from the spontaneous breakdown of acetoacetate and beta hydroxybutyrate, or are there other metabolites that create acetone?
That's the major one, and I would say I'm curious if it's actually just a spontaneous decarboxylation, that spontaneous breakdown, because there is some chemistry that shows the class of chemicals that acetoacetate falls into can be catalyzed by proteins. So the catalysis, meaning the speeding up the reaction of acetoacetate developing into acetone, can be done with a protein. And one of the proteins is, at least in my opinion, could be like albumin. Excuse me, and albumin is a protein that circulates in your blood. It's a large protein that circulates in the blood, and that could be providing a site for the conversion of acetoacetate into acetone. So it's not clear totally, but it does seem, like I said, there's some literature. There's actually a gentleman who did a dissertation in the late 70s that seems to indicate this could potentially be a another way. But to answer your question, acetoacetate is the main precursor or mother compound to acetone.
I see. So going back to the albumin, so there needs to be like a place for this decarboxylation to occur. It can't just happen like within the blood. It needs to be.
Yeah. So if it's spontaneous, it can happen anywhere. It can happen in the blood. It could happen in the liver cell where acetoacetate's formed. Yeah. Anything that has a watery medium could potentially make it happen. But if there's a protein that helps speed up the reaction, then once again, it could probably happen anywhere that there's a protein, but the blood is certainly, the albumin is present. There's lots of proteins in the cell that could potentially help that as well.
Interesting.
Yeah.
So maybe like low or high albumin would affect how much maybe acetone is made in the body.
That's a possibility. Now, this is all my hypothesis.
Sure.
There's no data. The reason I think this is possibility is if you look at the spontaneous rate of kind of decarboxylation, it really doesn't seem to favor spontaneity of decarboxylation. When you look at the kinetics and you look at the rates of these formations, and I've done that through some of my literature search. So it does seem like there's potentially something else that has to be helping this along. Yeah,
super interesting. I definitely want to dive deeper into that and how acetoacetate is formed and all that. But
right.
How I kind of got into this was through like looking at gut health and gut health research, and a lot of researchers are looking at other volatile organic acids in the breath. I think that's pretty fascinating. That like, in your opinion, do you think in 5-10 years, there will be more devices like Levl looking at different volatile compounds that will give us help us kind of triangulate to see what's going on inside the body? Is that kind of where?
Yeah, I think that can happen. I think one of the challenges will be, can you make sensors that can detect probably 1,000-fold lower concentration than what we're detecting with acetone. Acetone's parts per million. So a lot of the other compounds are parts per billion, or they're in that range. So probably more sensitive in terms of the concentration, as well as being able to differentiate the different compounds. So making sure that what you're reading as acetone is truly only acetone and not other compounds. Or if you're looking at isoprene or 2-propanol or whatever the other compound you want to look at, make sure it is that exact compound. So there can be some overlap.
I see.
So there's some more technical challenges. I'm never going to be one to say we can't overcome those, because those people always are shown to be incorrect. But there is still some more hurdles. But yeah, I definitely think that's a possibility, and that's going to be pretty amazing, because there's a lot of stuff in terms of science that we know can really benefit in terms of diagnostics and being able to understand your body better, and then being able to change and improve your health.
Yeah. Yep. What are some of the stories that you're hearing about from Levl users? Like, I mean, I know for me, stress, all that sort of stuff, but for like with fasting, that's where I really noticed like my breath acetone, even like for a day after, is much, much higher. Yeah. What are you guys hearing, like anecdotal stories from users and things like that?
Well, and the stories in terms of how they're using it, I guess, is one question or our answer. Is the stories I've heard. One of them is a gentleman who uses it to, control his type 2 diabetes, control his health and his brain function. And it's pretty interesting. He's an older gentleman, and he's like, yeah, I was interested in it, but once I started using it, I use it every day. It's transformed his life. Nice. And it's because he can constantly understand what he's doing and make sure it's improving kind of his health and wellness. So those are the stories that are probably the most exciting. Yeah. The other one that I always like to talk about is the mother-daughter who comes in. They're on the same diet, and the daughter blows 6 parts per million. As we know, that's elevated, and the mother blows like a 0.6 or 1. And the question is okay, you guys are doing the same thing, and they both vouch for each other. And then they start to have the conversation of, well, at nighttime when the daughter goes to bed, the mother rewards herself with a bunch of candy. Yeah. Right. And that just kills everything. So so that's 2 stories there, 2 kind of outcomes. Number one, we hope Levl allows you to have a conversation with people. Hey, this isn't working, and why isn't it working? Or a conversation of, hey, this is helping me achieve my goals, and it's helping me every day to get feedback to continue to improve. So it allows hopefully both of those conversations to occur. And I've also said this too, is Levl isn't necessarily, any reading is not necessarily good or bad. It's just simply where you're at. And don't worry about that. Worry about where you're going to go from there. So it's about moving forward as opposed to saying, oh, I messed up, or oh, I'm having a great day. Move forward and say, I'm going to use that information for improvement.
Yeah. And do you think I know you know a lot about psychology and stuff. Is that a key element for behavioral change in your estimation, like using these biometric and trackables? Because it seems like there's so many hurdles to change. Like human beings, we don't like new things. We like to go to the same restaurants, eat the same food, right? So seeing that data, do you think that helps to like catalyze that cycle of behavioral change?
And I will say I'm not any expert on psychology, but I think it does. I don't think it may not be for everyone, but I think a lot of people like to say, what did I do yesterday? Can I get away with that? I think that's one way, at least I think of myself, where am I at? Did I do too much to push me over the edge? Did I do too little? And I think the other thing is I like to have some feedback to say, am I doing the right things, right? But you've also got to temper that with, if you are not having success, don't beat yourself up. Just say, you know what? It didn't work out yesterday. Right. Today's a fresh day. Let's start over and let's move forward. So there is some people are fine with that, but some people can get down. And this is not something to beat yourself up over. It's really to say, is a starting point. Every day is kind of a new starting point, and let's move forward and improve. Yeah. Yep. Great point.
All right, let's talk about maybe defining the process of how acetone is made. We kind of tacitly talked about that a little bit. All right, so I'm fasting, I'm doing exercise, glucagon's high, insulin's low. I'm starting to liberate free fatty acids. Kind of what happens from there, and talk about the ketone metabolites and all that.
Sure. So the ketone bodies are created in the liver cell, and the fatty acids go into the blood. So you have fat stores, and everyone probably knows about those in their own body. But those get dumped into the blood circulates throughout the body, and some of those fats get put into the liver. And that's where the liver will use those fats either to create energy, or if you don't have a lot of sugar on board, they'll get diverted to create some of these bodies called ketone bodies. And before I explain that, one of the questions, at least I had, is why bother creating ketone bodies? It seems like kind of a waste of energy for the body to create these. You already have sugar. You already have the body should be able to use fat. Why additional chemicals? Well, I think historically our body has used those ketone bodies to feed the brain. The brain, for some reason, is unable to use fat for fuel. And it's not clear exactly why that is, whether it doesn't have the enzymes for it or whether the fat can't cross the blood-brain barrier. But it can't use them by themselves. So when you fast and you don't have sugar available, the brain needs something, and that's where the ketones come in as a substitute for sugar to feed the brain. So that's what's being created in the liver. So your fats, you're low on sugar, low on insulin, the fats are circulating, put into the liver cell. Liver cell creates some of those ketone bodies, so basically your body can feed your brain and fuel it. And so, like I said, some of those fats get diverted into a channel to create this mother chemical called acetoacetate. And then acetoacetate is the mother that can be interconverted, so back and forth, with beta-hydroxybutyrate. And I imagine a lot of you viewers probably know these names, are kind of big words. Or acetoacetate can be converted to acetone.
I see. And you referred to acetoacetate as the mother molecule. It seems like a lot of focus has been focused on beta-hydroxybutyrate. Do you feel that, and I know Stephen Finney talks a lot about this, that acetoacetate maybe has signaling properties that we don't fully appreciate? Like, is there more going on that behind the scenes that we are aware of, do you think, with that? Or?
Oh, absolutely. I think the reason beta-hydroxybutyrate gets all the attention is it's the one that gets measured. It's the one, it's kind of like the paparazzi takes pictures of this person, so they're in the media. They're not necessarily the one that's driving everything, but they're the one that everyone knows about. So we are measuring beta-hydroxybutyrate. So since you can measure it, you're going to learn a lot about it. But I do agree that acetoacetate is probably more important because it's a mother chemical. It's the chemical that actually it's a chemical that's put into the process to fuel the body. So when the body says, I've got to use ketone bodies for fuel, it takes beta-hydroxybutyrate and through conversion converts it into acetoacetate before, and then acetoacetate gets put into the fuel cycle to fuel the cell. So beta-hydroxybutyrate doesn't fuel the cell directly. It has to get converted to acetoacetate before it gets used for fuel. So acetoacetate really is the molecule That's kind of the pivotal molecule, but it's not really being measured.
Yeah.
Because you don't have these
Devices
consumer devices, and it's just a little bit more difficult. You can do it in the laboratory, but not for the consumer. And so that's why beta hydroxybutyrate has been the one to talk about.
Interesting. Now, is acetoacetate like stable or unstable? Is that why you can't measure it, or why?
No, it's certainly available and stable. And I'm not, I haven't looked into why it's more difficult to measure. But I imagine it could, once again, like we talked about before, it can be overcome. It's not a problem.
Yeah.
Yep.
Okay. So I have heard that hepatic output. So we're in ketosis, like you mentioned, low glucose, low insulin, et cetera.
Yeah.
Our body's liberating free fatty acids from fat tissue. Our liver's starting to make these ketones and so forth. Dr. Stephen Finney from some metabolic ward studies was, kind of talking to me. It was at a cocktail type party at a seminar, saying that 50%. So the BHB goes more towards the brain, and then the muscle tissue tends to utilize the acetoacetate. Have you seen any of that research or any feedback there?
Yeah, I haven't necessarily seen that. Acetoacetate, certainly there is some arm studies where acetoacetate gets used for fuel in the arms, and beta hydroxybutyrate, once it gets to the brain, though, it does through conversion through beta hydroxybutyrate dehydrogenase, gets converted to acetoacetate before it gets put into essentially the Krebs cycle and be used to generate energy. I think the one interesting thing for me is that a lot of the utilization of all these ketone bodies happens early in the transition between, your sugar burner and then you're changing over to ketosis. So you have this change, you have this big increase in beta hydroxy or in these ketone bodies during that conversion time. Once the body starts to ramp up the ability to use fat for fuel, then the ketone bodies start to drop, and now the ketone bodies are mainly there to fuel your brain, and then the fats are able to be used for muscle tissue, hearts, skeletal muscle, etc., and fuel those things.
Such a great point.
So there's a transition time, and that's what you see in a lot of people who've converted from using sugar to using fats. Will see a huge spike, the first few weeks, and they see it kind of drop off with time. And they shouldn't be scared about that. They should just understand that's the body's natural process of changing from using fats, sugars predominantly to using fats.
Yeah. So the mindset is, I want to be in ketosis. So
right.
They focus so much on that, but then they see that maybe ketone levels are going down. They think they're not burning fat for fuel, but they really are.
Right.
And would you argue that's really the goal, right? I mean, is it, people say fat fueled and they kind of confuse that with being keto. And obviously there's a lot of overlap there and things like that, but I think for a lot of people, that is the goal, to get your cells, mitochondria, more healthy, more active. So you're using fat for fuel. Yeah. And ketones are an offshoot of that, particularly for the brain, et cetera. Yeah. So it's an interesting, so how do you kind of separate those two ideas, like burning fat for fuel and beta oxidation and ketosis for people? Because that does kind of get confusing for some folks.
Yeah, and I think it depends on how you're deriving your ketosis. For example, if you're deriving ketosis by just simply nutrition, meaning I am going to cut back the sugars that I'm taking in, and so by virtue, I'm going to increase my fat load, and it's dietary ways of increasing ketone levels, versus you're supplementing, which is another way to do it, right? So that's a little bit different, and I think that's going to have different effects, and it's going to have different impacts on it. Yeah. Versus you're going to fast, which is obviously, now it's not dietary in a sense, it's maybe archaeologically dietary, but it's on you, versus I'm going to do calorie restriction. So there's lots of different ways, and I think for each one, there may be a different viewpoint on how you kind of separate, kind of like you said, the fat metabolism, the beta oxidation, from the utilization or the ketone levels. So I think each one of those kind of has a different way of thinking about it. Yeah. And we could certainly walk through that a little bit.
Sure.
Yeah.
Well, do you think there will ever be a point where we can measure, is it the non-esterified free fatty acids in the blood that ultimately go to that acetyl-CoA pool that eventually make the ketones? Is that where?
Yeah.
And so why can't we measure non-esterified free fatty acids like commercially?
That's a good question, and I don't know the answer to that. But certainly you could, because they measure them. You do a blood draw, and they measure them in the laboratory.
Right.
The question is why can you not measure them with a blood glucose meter and that sort of meter? I'm not sure. Yeah, I don't know that. You'd assume a guy who's got an engineering background would have an idea about that, but I don't.
Yeah, that would be pretty
I'll
interesting,
look into
right?
that. Yeah, I'll certainly look into why you can't just measure the fatty acids. And I will also say, the question is, just because you have a lot of circulating fatty acids doesn't mean does that mean you're metabolizing fat? I'm sure there's a way to figure that out, but
sure.
There is, just because they're circulating doesn't necessarily mean you're using them.
That's a good point. Yeah, I think I was reading a few studies with people with NASH and non-alcoholic steatohepatitis, fatty liver. Right. So they were looking at, I think it was log. So it was the ratio of, I think it was beta-hydroxybutyrate or acetoacetate to non-esterified fatty acids, and looking at citrate. So looking at the whole picture of the mitochondria and everything. So I know it gets complex, the biology, but I was just curious,
And what did that show?
Well, it could show kind of where the hiccup is. Is the problem coming from the free fatty acids into the liver? Is the problem within the liver, or is it even getting more into the minutia of the mitochondria? Is there like some hiccups there? And this research paper figured out that was pretty much it, because I can't remember, citrate was really high, and that would suggest that there were some downstream issues going on because ketones were low at the same time. I could be a little off on that.
Sure.
But yeah, I don't know. I thought it was super interesting.
Yeah, and I think you bring up an interesting point of, can you make multiple measurements to identify what's happening? I think there are people who are doing full blood panels to try to figure out, maybe monthly, to try to figure out exactly what's going on metabolically in their body. But you also bring up another point of the mitochondria, because the beta-hydroxybutyrate and the ketone bodies do seem to be able to bypass some defects in mitochondrial function. And when we're talking about defects in mitochondrial function, we're talking about the inability of the mitochondria to generate fuel from, like, for example, sugars. So if the mitochondria, which is your power plant of your cell, isn't able to generate fuel that the cell can use, ATP, from your primary fuel substrate, which is sugar, if it can't do that conversion sugar to ATP, then you've got to have something else to feed it, and that's where the ketone bodies can come in. And that's kind of the whole basis of how the ketone bodies can be therapeutic for brain disease, like Alzheimer's, Parkinson's, dementia, those sorts of things. That's kind of the hypothesis. Some of those have been demonstrated better than others, but there's more data to be had, so to speak.
Sure. Yeah. My friend Max Lugover has a great kind of analogy for that, like this energetic backdoor.
Yep.
So what you're talking about, kind of like the insulin resistance or mitochondrial dysfunction, and then the ketones can come in and kind of provide that alternative cellular energy, which I think is pretty fascinating.
Yeah, and there's, for example, for like dementia, one of the things that they've done with brain scans is shown that people with dementia may have regions of their brain that are not functioning, and not functioning in the sense that they're not burning sugar. And so when you can provide fuel for those parts of the brains that aren't burning sugar, that aren't basically non-functional, then they start to process again, because you now have overcome, like you said, gone through the back door and provided them fuel, so those cells can now kind of turn back on. And that's, once again, a little bit of the hypothesis of how ketosis, elevated ketosis, can help people with dementia.
Super interesting stuff. So you hit on something earlier that we didn't follow up on, and trying to ascertain or elucidate how their metabolism is more in a fat burning state. And so is that where acetone comes in? Because that gives us one way to triangulate and see if these things are being utilized. Is that kind of what we're saying?
Right. And so acetone, the way we've seen it, and I think we'd all agree, the folks at Levl as well, there's more data that needs to be gathered. But acetone really is an indicator of kind of your last 12 hours of fat metabolism. So, because there's a little bit of a delay between the changes in acetoacetate and beta-hydroxybutyrate in acetone, and that's been shown in a number of different studies. We've seen it too in our studies. But yes, it does kind of give you an indication of is your fat metabolism being elevated, and if it is, that'll be reflective in acetone. But like I said, it's more of an integrative over the last 12 hours or so.
I've noticed that the changes, like in glucose, are much more rapid than ketones and acetone. So why is it that 12 hours? Explain that a little bit more.
Well, for acetone, I think one reason for that is just the kinetics and some of that some of the way that acetoacetate changes into acetone, that reaction takes a little bit longer. So I think for the acetone levels to kind of build up to where the acetoacetate and beta hydroxybutyrate are, it just has this delay in the kinetics. And I think it's due to the slowness of the kinetics. So you've got to get those two molecules up to a certain level in the sense, and then it's got to have time for those to be converted, or acetoacetate to be converted, for it to then show up in a breath measurement.
Interesting.
Yeah.
I found it pretty fascinating. I mean, there is real-time changes, obviously, right, with the breath, and I think it's a little bit more fast than using blood.
Right.
So I've been tracking, I have this continuous glucose monitor. I have like the blood, and just to try and experiment, just to see like what changes, how much carbohydrates can I have around exercise,
right,
so as to optimize exercise performance, but still kind of burn fat for fuel, primarily for brain health, longevity. And so using these different modalities, it's been really fun. And so that's why I like, the Levl app. The first night I got it, I charged it and like got it all synced and stuff like that. And I had a dinner party with a bunch of low carb friends. So we were all like had it out, and some of us, including myself, were having some wine and everything like that. So let's talk about like, how does alcohol affect? Maybe does it affect the sensor or the reading, or?
Yeah. So there's a couple different things with alcohol. Alcohol affects the sensor, and the sensor measures the oxygen group. It measures a sensor basically is it's a measurement of oxygen on and off the sensor, and the acetone can kind of push that oxygen on and off the sensor. When I mean oxygen, I just mean oxygen in the air.
Okay.
And so there's kind of this changing in oxygen that the sensor is measuring, and alcohol has oxygen as part of the molecule as well, and so it can mistake alcohol for acetone. So normally most people, if you don't have any alcohol in your system, it's small enough, it's parts per billion level, it's 1,000 times lower, so it's not going to affect the measurement of acetone. But if you drink alcohol, you can get a few hundred parts per million, so you can get 300 parts per million or so in your breath, and then that just swamps out the sensor. The other thing that alcohol does too is, let's say you've drank alcohol and you've waited 8 hours, and so the alcohol's out of your system. You can get this little alcoholic ketosis that you can see with acetone, that's due to the alcohol, the way the alcohol is processed in the body. And so you can see a little bit of that. Typically you see that while the alcohol is being burned off, and you have to have something that's more specific for acetone. And we see that with our mass spectrometer in the laboratory. But most people aren't going to see that. Most people, when you breathe into this, you're going to see it give you some ridiculous number, and that's just due to measurement of alcohol.
I see. So the alcohol ketosis, that's like the ethanol breaking down in the body. It stimulates ketosis, or it looks like ketones? Or? So
when it breaks down, some of the intermediate molecules, the energy molecules, can help convert acetoacetate to beta hydroxybutyrate and start that process. And then you see some of that show up with acetone.
Interesting.
Yeah. So you get a little bit of ketosis, and that's also a little bit of the reason why you get this fatty liver disease, is some of the alcohol is driving that fat metabolism. Yeah. So that's where you see that, and you can see it in the acetone measurement as well.
Interesting.
Yeah.
I wonder if fructose has a similar effect, 'cause I know that fructose stimulates kind of the lipogenic pathways. Have you looked at that, fructose? And that?
I have not. Yeah. Fructose is an interesting one. One of the things that's interesting about fructose is just the fact it can be absorbed more readily by your gut. So it's a great sugar to use when you're exercising. Yeah. Because you can if you're in a long-term mode of exercise, you're doing a marathon or something, if you eat fructose, it can get into your blood, and you can start processing a little bit faster than sucrose or some of the other sugars. But I haven't looked at it in terms of the other piece.
No, the ketones and everything. Yeah, I used to read a lot about fructose and all that when I was racing, Cat 2 road cycling and stuff like that, because a lot of the intra workouts or some of the fuels, it was like back then it was popular. It was Hammer. I don't know if you do any of these athletics, but yeah, so it was like dextrose, fructose, and there's all these weird sugar molecules because they don't affect the GI tract as much. So if you're exercising, right, you don't want a lot of stuff sitting in your gut and can cause issues. And so I found that, and it was like the osmolality, I think, of that was part of it.
That's part of it. Did you guys use fructose? Was that something that was heavily used, or not?
It was promoted, in the bodybuilding world too. It's a thing right now because of how it stimulates the liver in these different pathways. So
interesting.
Yeah,
I'll have to take a look in that. We've typically kind of stayed away from, in terms of the research side, of kind of the glucose and the sugars, because that obviously elevates insulin, and that kind of shuts down ketosis. But
Right.
but that doesn't mean it's always going to shut it down. I mean, I'm sure there's some situations where you may be able to overcome it. But I'm not as familiar. Yeah.
Yeah.
As familiar.
Interesting. What do you do for exercise personally?
Yeah. So for me, I used to run a lot. So I used to run 4 or 5 miles, probably 4 times a week.
Yeah.
Because that's kind of my background, more of a runner. But more recently, I've kind of transitioned to doing more push-ups and pull-ups along with running, kind of all together. So I'll run for a little bit, then stop and do push-ups and pull-ups and run some more and do the same thing. And I actually have felt a lot better doing that than running just by itself. And it's and I do it 3 or 4 times a week, that sort of thing. Yeah.
Yeah, I find that mixing in that resistance training with the aerobics is, a lot of people just get stuck doing one or the other.
Yeah.
And recently I kind of have scaled back just from time working on the house and everything like that. We do a lot of hiking, and I find that like doing prolonged aerobics like that really helps to kickstart some of these fat burning, type things. So we did a very long hike a couple months ago, and my breath acetone was like off the charts, for that. So yeah, fasting, obviously low carb, high fat diet helps with that, but I like to get into ketosis through exercise or fasting, and diet's great, but
and that's probably a perfect. There's a nice study showing people who went on these, they have like these 3-day walks that they're walk for a charity, and they did one of these in Belgium or the Netherlands. There's a study that showed that the people who walked for 6 hours a day would have really elevated breath acetone. And that's probably what you saw with hiking.
Yeah.
Whereas the other thing that we've seen, kind of the opposite, is we have folks at Levl who are on ketogenic diets, but then they're going to do a soul cycle class, so a hard cycle class for 45 minutes where they're all out, and the acetones will drop. As a matter of fact, their blood ketones will drop too. And a lot of that's just because they're burning
using the fuel. Yeah,
they're using it for fuel. And I think the way that acetone's stored in the body, it's maybe a little more transient. I mean, you can probably exhale a lot more of it, or you can metabolize it faster, and so they drop down. And then what we typically see too is in 4 or 5 hours they're coming right back up.
But at first they're probably confused, like, wait, I got kicked out of ketosis. But it's like, no, you use that energy
for fuel.
Yeah. So that's kind of the thing to mentally wrap people around, because I see that a lot. Like people will go to the gym, similar thing, use like, just beta hydroxybutyrate, blood test, and they get really confused. So I'm glad you hit on that, because
and that's
confusing.
And that's the other thing for the Levl device. Obviously, there's lots of utilization for it, but that's the way you kind of say it's a 12-hour measurement. And that doesn't mean you can't measure every hour. You can measure, every time it's available to do the measurement. But it does allow people not to get too, too focused on it and wrap their, get so focused that they kind of lose track of the end goal of it. Yeah.
Well, and it's non-invasive.
Right.
It's not expensive. Like, every time you do, whether it's Precision Extra or Keto Mojo, it's like $1.25 or more, right? So it's like, this is nice because you can test it all, it doesn't cost you any money.
Right. Nice. And you can start to get a sense of how your body's going to respond to different things. So if you do some activity or you do some eating, like I said, there may be a little bit of delay on the nutrition side, but certainly on the activity side, it seems to be a little more congruent with whatever activity you're doing.
Yeah. Okay. So on the app that syncs with your iPhone and all that, which is great, it's Bluetooth. There's some ranges you guys have. I think it starts at like 4.
Yeah, the range for the breath acetone measurements. So we go down to 0.5 part per million is kind of the level of sensitivity, and then kind of every 1 part per million. So 1, 2, 3, 4, 5, 6 is what we go up to, and then after 6, if you want to see the reading, it's on your app.
Yeah.
So you can see it on the front of the device. It'll show 1 through 6. Then the rest of them are on their app.
Okay.
And anything in between those numbers, obviously we go to tenths of parts per million, so you can see that in the app as well.
I see. Yep. Yeah. And I think like over 4, it tells you how much fat you're burning per day and all that sort of stuff. So I'm curious is like how that was calculated. How did you guys kind of figure that out?
And that's and I think that's something that we try to emphasize, we try to explain. So that calculation was from a study that was done in the early 90s by Abbott Laboratories, and they put people on a calorie restricted diet. So obviously you kind of have to fall within the criteria of what happened. So these were folks on calorie restricted diet. They weren't on high fat. They were kind of a mixed diet, but they just cut back their calories. And then over the second half of the study, they showed that people who had certain body fat losses had a correlation with the breath acetone.
Yeah.
So 2 parts per million is what they showed would give you about a half pound of fat mass loss. So not body weight, but fat mass loss. If you went up to 4, it kind of doubled. It's not exact, but that kind of gives you a 1 pound of fat mass loss at a 4 parts per million. And their range extended around 10 to 10 parts per million. So one of the things you'll see on the app is if you get 12 parts per million or 20 parts per million, you won't see a fat calculation.
Right.
And the reason is because that's kind of outside the data range that was for that. But the fat mass calculation really is more for people on a calorie restricted mixed diet. So if you're on a calorie restricted, but you're on a ketogenic diet, which is great, you're probably going to be, most likely you're going to be above 10.
Burning more fat. Okay.
Yeah. Because you're going to be burning fat because you're fueling your body with fat. I mean, you're ingesting fat as well as you're pulling fat from your body stores. So most of the energy your body's creating is from the fat sources.
Yeah.
Yep.
Super interesting. Yeah. So that gives people a nice metric because they're unfamiliar with acetone, right? They know like pounds and everything like that.
Yeah.
And then once they get it into, it took me, to be honest, about a month to really kind of know where things would be at because you're familiar with glucose and other things. So it's nice like to learn that and then figure out, okay, well, if I fast or if I exercise or I eat carbs, like this generally happens. But I noticed, like I said, fasting and exercise, it goes like on the parts per million, like 35 and higher sometimes. I think one time, and I recalibrated the sensor and everything. So I'm like, it was like over 70 one time. Because I did like 3-day fast. And I was like, holy crap. I'm like, this can't be right. And I like redid everything. So
yeah, and that's not surprising. We see people who, like for example, if you were fasting or you're on a very strict low sugar diet, and then let's say you supplemented with one of
ketones
the ketone esters or the ketone salts, you can really crank it up quite a bit. And I'm not saying that's necessarily what you need to do.
Sure.
There may be some utility for that for different disease and therapeutics, but yeah, that can certainly happen with fasting. And once again, it goes back to what I said before. When you're in the transitionary state, your body's going to be overproducing ketone bodies in a sense because the rest of your body, your skeletal muscle and your heart muscle, hasn't developed the infrastructure, the machinery to use fats. So once it starts to ramp those up and realize, oh my gosh, I'm not going to be fed with sugar anymore, I've got to have the machinery to burn fats. Once that machinery gets ramped up, then the ketone bodies will fall off just so they can feed have fuel for your brain.
Yeah.
So that's probably what you're seeing. You're a little bit in that transition zone. If you were to do a longer term fast, it'd probably drop off a little bit. You know, if you're doing a 30-day fast, I'm not recommending it per se, but you're gonna go up, and then it's gonna drop off a little bit. And they've shown that quite a few times.
I see.
Yeah.
So the body's trying to, because it seems almost like a waste of energy.
That's exactly it. Because otherwise, if you're not going to use those fuels, then you're going to excrete them usually through the urine. You're gonna excrete those, and so the body's just wasting. It's not being efficient.
Sure. So it's initially, and then over time, it realizes, hey, we're burning here.
Yeah, it realizes, it says, oh, now all my cells have ramped up their ability to use fats for fuel. There's no reason to have this intermediate molecule, so it'll cut it back. You still have to have it for brain fuel, obviously. And so then once you get farther out, there's the idea of that perhaps the amount of ketones concentration are a little more tied to your brain to body weight ratio. So smaller people, the brain size is pretty constant in a lot of cases. So, smaller women may have higher concentrations than bigger men because the amount of water that those ketone bodies have to be dumped into is bigger for a man. So it's a lower concentration versus a woman. It's smaller volume of water. So it's a higher concentration, even though they have the same brain size.
Interesting. I've never heard that before.
Yeah,
that makes so much sense, though.
Yeah, it's kind of if it's being used for fuel for the brain, then that's kind of where that concentration. But that's a longer term. If you were to do a 30-day fast, you may start to see some differential there. And there's a gentleman who's passed away, but he did a lot of this fundamental research, and I'm forgetting his name at the moment. Oh my gosh, I can see his
Cahill.
Cahill, yeah. And he had a paper where he kind of showed that children typically would have higher concentrations than women than men, and you'd have that kind of order, and that makes sense for children, right? Because they have the brain to body ratio is smaller, or is higher than you would for a man.
That is really, really fascinating. So, do other animals utilize ketones? Is that kind of energetic backdoor too?
Yeah, and the one that, for example, I'll go back to Cahill. The one that he talks about is like the bear. So the bear goes into hibernation. So the bear will use, and I think they've pulled some of these. I wouldn't want to be the guy doing this, but they pulled the bear out of hibernation
Yeah.
and done some measurements on them.
Wow.
But they're used ketones, and that makes sense because they're using the stored body fat. So some of those are going to be used for the brain as well. Yeah.
I've heard that dogs don't really make much appreciable amounts of ketones. Do you know anything about that?
And I don't know that. Yeah, I'm not sure. But I know every animal is a little bit different. For example, cows make ketone bodies, and there's some concern for that in the kind of the agricultural world, because those ketone bodies, the ketone acids, will flavor the milk differently. So they try to not allow them to get into ketosis too far, because it'll change the production of the milk.
Like more wheat here, eat more
Right?
carbs.
Yeah, it changes the taste of the milk, I believe, and I think I'm right on that. I could be wrong. But so there are different animals. Have different propensities, in my understanding, but I haven't looked too much into it.
Yeah, it's pretty fascinating. I mean, that's big money that they're talking about. I mean, even there's this whole thing. I don't know how I got into this. This whole notion of pre-slaughter stress, and these animals, like if they get transported across 3 states to be processed, they get dehydrated. There's that perceived stress, and all that sort of stuff, and that changes the acidity of the meat, and makes it much less palatable and more acidic and everything like that. And so, the acid balance. I mean, so. When you're keto, does it really affect like blood acid to base balance? I mean, are they that acidic?
Yeah, and that one I've looked at some. I don't think it does, but I am by no means the expert on that.
Sure.
But I don't think it does, because a lot of when you look at the number of hydrogen atoms, which is really driving the acid-base balance, the ketone bodies aren't producing that many, because there's not so many of them. I think it certainly will if you get into ketoacidosis. So if you get into a disease state associated with diabetes and it's uncontrolled production of ketone bodies, that will certainly help change it. But I think the levels we're talking about are small enough where we're not really going to change what we call the pH, the acid-base balance within the blood.
Yeah. Interesting. So the first time I ever heard of ketosis was in 2002. It was because I was working in a gym in Gold's Gym. I was just there this last week, actually, in California. And one of the trainers was on a ketogenic diet. And I knew, like, every time he would come in, his breath stunk. And I was like, what is this guy? He reeks. And so part of it could be like protein malnutrition. Like, he's overdoing the protein. It's putrefying, and that's part of it. But he was really a diehard keto guy, way ahead of his time. This was 2002. And so I remember in my head, I'm like, I never want to like have my breath smell like that. I never want to do this thing that he was doing, even though he was like the most ripped, and he did a lot of shows and stuff like that in Northern California. But that smell, is that the acetone? Yeah,
that's typically the acetone. Now, you could have a little bit of ammonia too that could be associated with, but I think it's mostly acetone. And usually you can mask that with gum or something along those lines.
Sure.
Too. Yeah.
But it's something I've noticed, like my keto friends are a little bit like almost sweet smelling. Like,
yeah. And that's typically, people who were diabetic back, I mean, they always talk about this, the academic literature. Back in the Greek age, they could smell it on their breath because it smells sweet. In fact, sometimes it smells a little bit like alcohol, and it can be mistaken for alcohol in the breath. So if you don't have a good sense of smell, you can say, oh, you must be drinking alcohol, and it couldn't be ketosis.
Interesting.
You're not having as much fun as you should, apparently.
Yeah. Now, does acetone come through the sweat too?
Yeah, it can. Certainly. Okay. As a matter of fact, there, once again, nice study that shows they can measure it across the skin, but it gets delayed even farther across the skin because you got to go through the skin barrier.
I see. So it's not, the quantities aren't appreciable, but it does happen.
Yeah, a couple things. So the quantity, the concentration will be less than what's in the breath. And then if you were to compare it to the breath, it's going to appear on the skin later. It's going to have a delay in time with the appearance in the breath. So the breath spikes. That spike won't be seen for maybe another few hours across the skin.
I see, by the time it gets there. Okay, so that's why breath is good. Now, what about urine? Is this spike similar in breath and urine, or?
No, and the problem with the urine, and it's probably not worth going through it too much, but the problem with the urine is if you get dehydrated, your urine output decreases. You're dependent on how often you need to go to the bathroom, and so the urine is collected in the bladder. So over time, you're just having this mixed concentration. So it's not really a good measure. The other thing that's interestingly a problem with urine is that acetoacetate's in the urine as well. And it's the mother compound for acetone.
Yeah.
So if it spontaneously decarboxylates, you're creating more acetone in your urine. So that can also throw out the measurement in terms of a high concentration.
False positive.
Yeah, in a sense. So urine's really not the best one to do. Now, it can be useful for some applications, but if you're willing to be more quantitative about it, it's just not a great marker.
Yeah. I think some people, when they first hear about ketosis, they go to Walgreens and they buy this and they, they kind of get into it that way. But I don't know anyone that's pretty serious about this that uses urine anymore. Yeah. So I agree with you on that.
Yeah, exactly. And to kind of go back to the quantity, one thing I wanted to kind of point out is, quantitation I think is nice, and to keep records of that so you can go back and see that. One of the things we talked about is the concentration of acetone with relation to fat metabolism.
Yeah.
Right. And one of the other things we've been working on and have kind of got a pretty good handle on is what is the concentration of acetone that would be comparable to the nutritional ketosis, right? The nutritional ketosis is typically defined as 0.5 millimolar of beta hydroxybutyrate from a blood sample. And so there's obviously a conversation of, well, how much acetone would be comparable to that? And we did a study that we finished that kind of shows 8 parts per million would give you, or higher, would give you at least a 0.5 millimolar beta hydroxybutyrate, because we did a breath to blood concentration. And interestingly enough, then we went back to the study that we published in 2015, have a bunch of data in there.
Yeah.
And that shows the same correlation, but that one showed it to be about 7 parts per million.
Okay.
Of breath acetone would be comparable, or higher would be comparable to 0.5 beta hydroxybutyrate. So right within the same range.
Yeah.
And there's reasons for why there's a little bit of a discrepancy. But yeah, so that's what you'd want if you want to get in that nutritional ketosis.
So around 0.7
7
or
parts per
higher
million.
is roughly 0.5 millimolar BHB.
Yep, that's what you'd see if you do a breath sample and you compare it with a simultaneous blood sample, you'd see that correlation.
That's awesome. Now, time of day, does it matter? I mean, you said given that this is kind of like a long term, like the 12-hour window, kind of represents like what you've done in the last 12 hours ish. Should you focus? Walk us through that. So if you putting that in context of the 7 parts per million or higher, there might be some ebb and flows in there. So just keep that in mind. Is basically the take home.
Certainly, will be ebbs and flows. You're absolutely right. I think the one thing to do, we recommend, and I will continue to do this, is to measure it in the morning, kind of the first thing when you get up. And the reason is you haven't eaten anything, so your body's allowed to kind of come to a new state. You haven't put anything in your mouth, so you're not going to contaminate the sample. And it's the same time of day. So if you can do it every day in the morning, then you have a marker to say, oh, how do I compare it versus yesterday? How am I going to compare it to tomorrow? With that said, let's say you can't do it in the morning for whatever reason. You get up too early and you got to get out the door. I would say try to do it one time a day consistently. That doesn't mean you can't do it other times, but have one time a day where maybe it's at lunch or maybe it's at dinner, where it's a consistent measurement. And so you have that same, you're not going to confound your measurement, or you're not going to contaminate your measurement by time.
Yeah.
I think the other time maybe to do it would be somewhere around dinner time, because you kind of have the same thing going on. Maybe right before dinner could be a nice measurement point, or right before you go to bed would be a nice measurement point to try to control for those other time factors. But yeah, you're going to have ebbs and flows throughout the day as a result of the way your body's, if you eat food, your body's going to change the composition of the breath, and it may dip and it may come back up. So you kind of have to keep those in mind. Or if you do exercise in the afternoon or in the morning, that's going to alter it as well.
Totally.
Yep.
I love that approach. So basically, like, be consistent, whatever that consistency is for you. Pick a time. Yeah. Because otherwise, you're comparing apples to oranges, kind of thing.
By virtue, that time variable is important, and that can throw you off, and it can make something that's not real appear to be real.
Totally.
Yeah.
But along those same lines, the cool part is it's non-invasive. You can retest. So to see meal to meal changes, like are these mangoes, or is this ketone product, or whatever that thing that you're trying to experiment with, is that influencing your body's fat metabolism or not? So that's why I like this, because there were some things that I was doing that was like, oh, maybe I shouldn't do that as much, or whatever, right? So kombucha, for example. My daughter loves dried mangoes, and I would have some of those, and that was causing adverse changes. But just knowing that, right, was just kind of interesting to see, because mangoes don't affect my glucose, but they affect acetoacetate and acetone output. So kind of interesting.
Yeah. So you saw a drop in your ketosis as a result of the mangoes,
right?
Yep.
But glucose stays constant. So it was like, wow, that's really interesting biology.
Yeah, and that was the thing with. There's been some people talking about niacin. I think it's vitamin B3. Taking supplements of that and how it alters. It seems, and there's been literature on this for a long time, but niacin kind of blocks fat, the breakdown of fat at the fat cell. So your fat cell doesn't dump fat into the bloodstream. So it's kind of blocking the ability of fat to just get in the bloodstream, and as a result, you have a decreased production of ketone bodies. So
interesting.
And it's transient, maybe just a couple hours, and then it comes back up once the niacin wears off. And the same thing with aspirin, salicylic acid. Will do the same thing as niacin, but I don't believe it's as strong as an effect as niacin, but the same sort of thing. A couple hours and then it wears off.
Wow.
So there are things that as you test your body, and some people may find that they don't have that response to it, right?
Sure.
But probably the majority of the population will have that response. But there may be some people who say, like you said, mangoes doesn't change my blood sugar, but it changes my fat metabolism. Or other people may not have that reaction. So it's part of it's to try to dial in what's working for you and try to figure out what part of your genetics, which we don't really know a lot about. Even if you go get your 23andMe genetic testing and you get all the letters printed out and thousands of pieces of paper, it's hard to make sense of. Totally. And we're still in the early ages of trying to sort that out. So just doing this empirical testing can help you kind of move through some of that without waiting for 20 years to figure out what this means.
Because those polymorphisms, I have a buddy who's actually creating a product that's going to compete with 23andMe. It's going to be more, robust. He's local. His name's Ben Lynch. And I was feeding him all the different, SCOT and all the different ketolytic enzymes that help us utilize ketones. But they didn't have enough variants, at least known variants, to test for, to make a DNA testing chip. And so some of those, he put in a few, but some of them we couldn't. And I was really disappointed about that.
Along those lines, there's a disease called beta ketothiolase, and it's one of the enzymes that helps the process, the ketolytic process, or I think ketone production. And if you don't have that enzyme, I think if you don't have the enzyme, you have elevated ketone bodies. I'm pretty sure that's the way it goes. Couple things about that. Number one is that the prevalence of that in the population is just almost zero. When you're looking at 7 billion people, I think the number of reported cases are it's a handful. Yeah. So how are you going to find that out on a genetic screen with, you maybe have 2,000, 30,000 different genetics profiles? It's not going to show up, right? Yeah. But the problem is it is real, and some people are going to have that. Yeah. And some people may or may not know they have that issue. Most people have it. We're certainly going to know it. But how are you going to be able to then say, I want to put my genetic, my whole genome into their database so that they can start to analyze it? Yeah, it's probably not going to show up. Yeah. And there's quite a few of those diseases that are kind of rare diseases, but they certainly have an impact. But how are you going to be able to get enough of them for the signal to come above the noise?
Totally. Yeah. That's a really good point. Along those lines, I've done some research into what polymorphisms we know about in fat metabolism pathways, ketone synthesis and ketolysis. And it seems that individuals from Saudi Arabian descent and Portuguese descent, the HMG-CoA synthetase, is that the main rate limiting? I can't remember.
And I forget that at the time. I forget which one it is, but it's certainly one of them in the pathway. Yeah.
It's kind of upstream. Yep. Anyway, those individuals from Portuguese descent and Saudi Arabian descent tend to have a sluggish variant of this. So getting into ketosis
is
for them at least. Yeah. So it's like, again, what are the implications of that? Should they not prolong fast, or should they rely more upon, MCT oil that's very ketogenic in nature, very easy? Because I think the longer chain fats take a little bit more cleavage and stuff like that, right? Yep. Is that part of it?
Yeah, what they typically show is the shorter chain fats, at least when you ingest them, can turn into ketone bodies very quickly. Where the longer chain, when you ingest them, or take longer. So, I don't know what I wouldn't. I haven't thought through that, but what would you use for someone who has that limitation, that rate limiting step? Yeah, I don't know at the time, but that's a good point. And I think just what you're talking about, as we learn more and more what populations have these different variants and what populations are more susceptible to different either foods or proteins or micronutrients, how those affect their nutrition, how those affect their body, then we're going to be a lot closer to understanding how to treat our bodies individually and get them the best performance out of them.
Yeah.
But there's a lot to do. That's pretty interesting stuff you just found out.
Yeah, I was interested. There was a genetic summit, and I want to contribute information from a ketogenic standpoint. So I was doing all this research into that last year. James Maskell put that on. But along those lines, are you guys like anonymizing data and starting to collect? So with all these people breathing into the meter, they got this app. I mean, it seems like a great opportunity to learn from like an open source standpoint. Are you guys?
Yeah, we haven't done that. No, we look at the data in a gross sense to say how many people are using it, how often they're using it in that sense. But we really haven't done anything with individual data, and we wouldn't do that unless we asked people to opt in. And we would obviously do anonymize the data and de-identify it and that sort of thing. There are some other folks that we have partnered with who have our app. Heads Up Health in particular has them. And so if you have data from blood pressure, weight, body fat, et cetera, and you put it into their app, and then you tie our measurement in, then you have this whole body of data that then could be analyzed in a
that would be cool.
Yeah, in a more holistic way, and there's quite a few people doing that. But I don't know if they, because we don't have control over that.
Right.
I don't know if Heads Up Health has asked people to kind of opt into more of a study to do a more in-depth study or not, but that would be a great opportunity to start to learn even more.
Totally.
Yeah.
To start to correlate, make different correlations. And I wonder if the Oura Ring, do you know if that feeds into their API too? Heads Up Health?
I don't know, but I imagine they would like it too, because they're starting to get a big, nice database keeping track of things. But I would be surprised if they're not interested in trying to get that in as a part of it.
Do you know those guys, or do you know if Brett knows them?
I do. Yeah. David Heads Up Health. I did a little. I think he and I had a podcast.
Cool.
A little while ago. Yeah, they've got, it's a pretty cool product in terms of it stores all your data, and then you have historical. And the nice thing too is it tracks and trends it. So if you want plots, you want to compare something that people normally don't compare. Maybe it's blood pressure versus breath acetone. You can do that comparison and plot it on their device in their app.
That's awesome. Yeah, the Oura Ring allows you to do that on their cloud too. So you can look at heart rate variability in sleep, or look at heart rate variability in like resting, what time you have the lowest heart pulse during the night, stuff like that. There's all these different things you can correlate.
Very cool.
So that's cool, Joe. We talked a lot about ketone metabolism, ketone testing, fatty acid oxidation. Anything that we missed that you want to let people know about?
No, I think that kind of covers it. Like I said, I think the Levl app is great. The Levl device is great because it does allow you to get all that feedback. Totally. It allows you to track and trend and make modifications, make changes that are going to improve your health and wellness.
Yeah. So there's a couple different options. Like if you're a health coach or a doctor and whatever, there's that tier where you can have a lot of the users and everything like that. And then like I just have I think it's $50 a month, for me. Yeah. What are those different, options? I mean, they're on the website too, but like, what do you recommend?
Sure. If you're an individual user, you can buy one for your home, and allows you and someone else in your home basically to use it. You have a kind of a Jack and Jill. You have a his and hers breath pod, and then you can have two apps connected to it. And it's pretty easy. The breathing pattern. Yeah. What our goal is to have an easy breathing pattern: deep breath in, hold it for 5 seconds, big exhalation through the pod. But the goal there is to have a breathing pattern that's comfortable, that you can do every single time. So
I see. Consistent.
The breathing pattern is an issue. Now, if you have, let's say you're running a gym or you're running a clinic, and you have quite a few different clients, you can have one device and all your clients can use that. And then all that information goes to the cloud, and you have the ability to review all your clients in one database. And so you can say, Jenny came in and Phil came in and they're doing great except for on Thursdays or Wednesdays, and you can have conversations around what's working, what's not working, and how to modify and change those behaviors. And that one, like you said, that one has a different price point per month.
I see. But so the client would have their own app that would sync with Bluetooth.
Yep.
But that the owner of that would be able to see all the data.
Yeah, and the client has to say, okay, I'm going to share this with you.
Sure.
So the client may have their own, or a lot of times the gym will have its own app, and the client, there's a list of clients. So you just hit your name, you type your name in, and then you type in your password, and then it connects. And so the next reading that's made will be tagged to your account, and then that gets sent over to the cloud. And once it's to the cloud, you get it on your app, but your health coach would get it on theirs, provided you said it's okay for them to see it.
Give them the access, yeah. And then so you called them a straw? So that if you have like people over and you want to test and everything, so the guest, I love that because like people see it. I mean, it's a beautiful device. You guys did a great job with that. Right. Having it on the counter nearby so it's convenient, but then testing your friends and stuff like that. So I really enjoyed that.
Yeah, each person, they look like we call them mouthpieces. They're just paper. They're just, like you said, they're paper straws, but they're mouthpieces. And the idea is to be hygienic.
Yeah.
So not everyone's blowing through the same one. Now, if it's you and your wife, maybe that's okay.
Sure.
Or you and your partner, whoever it is. But yeah, if you have a bunch of people over, it's more fun to keep it clean.
Yeah, that's cool. Awesome, Joe. So it's levl.levelnow.com.
Levlnow, LEVLNOW.com. Yep.
Okay,
yep.
Cool. And then if they use if anyone listening right now or watching uses promo code HIH or levelnow.com/HIH, they can save $50.
Oh, awesome.
On the app. So we got that set up. I like it. And then so let's maybe just finish off on the new calibration canisters. Those are what we're going to be getting used to in the next months to come, right?
Yeah, in the next months we'll probably transition back to something that's more of an aerosol. But yeah, the larger canisters hold a lot of calibrated concentration of acetone. And it just allows you won't have to switch those out for probably a few months. I mean, probably close to half of a year.
Nice.
The only issue with those is you have a little bit of what do you call that?
The adapter piece.
The adapter piece, right? That you have to screw on, and after you use it, you want to unscrew it because there's a small chance if you're not careful, some gas could leak out, and your whole canister could drain with time. Right. So it's pretty easy to screw it on, screw it off.
And it's not volatile; just you need it to calibrate, right?
Yeah, exactly. Yeah, the idea is the device is a sensor, and that sensor can change a little bit with time. So you do want to just make sure that it's as close as possible to accuracy, and that's providing the accuracy.
Yeah.
It's like a scale. Scales can say, hey, you're off, and you can find out later. I put on a known poundage and it didn't, it's like, it's weighing.
I'm 220. What the heck?
Yeah. And then, but it could be like you put on a 100-pound weight and it actually says only 75. You say, oh man, I'm actually 300 pounds.
Right. Right. That's a really good point. Yeah. Cool, Joe. Well, thanks for coming on. And if people want to connect with you have a few articles that you published.
Yep.
Those on the Levl site, or should they look in PubMed? Or
so we have an article that is peer-reviewed that's on PubMed, but they're on the Levl site as well. And then we have some different blogs about utilization of Levl for different either weight loss, disease therapies, and different applications as well.
Yeah.
So you can look at levelnow.com has those as well.
All those, and then maybe just since we didn't really talk about it, I mean, it's tacitly implied that keto can be used for cancer, autoimmunity, inflammation. We talked about cognitive improvement. Yeah. What are the best applications? Like if someone's listening and driving right now, who would likely benefit most from one of these?
Well, I think the first application was weight loss, so just calorie restricted weight loss. Even if you're not on a ketogenic diet, I mean, I think the ketogenic diet has some benefits, so that would be probably preferred.
Yeah.
But just simply weight loss. Hey, I want to lose a few pounds. I want to make sure I'm starting to go in the right direction. That's one. If you are moving into a ketogenic diet, how can you watch your body and make sure that if you cheat on a day or two, how to make sure you're getting back on track? So I think that's a nice application. There's some other a little more niche. For example, type 2 diabetes reversal. There's a company that's reversing type 2, and we've known this. Go on a ketogenic diet, but one of the things you want to do is make sure you're on that in that state of ketosis. So I think those are brain function. I think that's maybe the other application too. People want to improve their brain function. Ketogenic diet's going to help you, but how to make sure you're in a therapeutic dose of ketones. And that would once again be the nutritional ketosis, which is on the Levl device is about 7 parts per million or higher. Gets you that improvement in brain function.
Yeah, that's a great tip. I didn't know where that kind of threshold was. I'm glad you told me that.
Yeah, and I should say I should back off that a little bit. Yeah. Levl is only indicated to measure acetone, so we're not indicated to demonstrate any sort of therapeutic benefit. So what I'm talking about is with respect to the literature. Literature seems to indicate that if you get into a state of nutritional ketosis, it should improve perhaps brain function, diminish some of the dementia, that sort of thing. But Levl can help you understand where you're at.
Great caveat.
Yep.
Love it. Cool, Joe. Appreciate you coming on.
Absolutely.
Have a good one.
Yeah, you got it.